my name is Cheyenne. I am a senior biology major at Howard University, and I had the opportunity to intern at the University of North Carolina at Chapel Hill in Dr. Haygood's lab, and he specializes in lung repair. So the goal of my project, well, what I was doing is I was comparing KRT5 negative basal cells in distal and proximal airways. Okay, so a little bit of background first. So breathing air in general depends on the efficiency of the lung's ability to transfer oxygen and carbon dioxide in order for oxidative metabolism to occur. So the anatomy of the lungs in general, so the respiratory system is like branching. So it stems from the trachea, and then you go to the bronchi and the bronchioles, and ultimately leads to the alveoli, which is where gas exchange takes place. So the lung is made up of a bunch of different types of cells, and each cell has a different function depending on where it's at or where it's localized. Okay. So the mature human lung is lined with what we call a pseudostratified epithelium, and it's made up of a bunch of different cells. So it contains luminal airway cells, which work to remove various inhaled particles via mucus. So as we can see here, there's a bunch of different types of cells, but we're focusing on the basal cell here. So the basal cells line the epithelium, and they kind of act as a stem cell. So in order to maintain homeostasis, they can self-regenerate, and then they can also differentiate into various different cell types as a result of some sort of acute injury or a sickness. So when we talk about proximal and distal, so the trachea repeatedly branches from the main airway, which leads to the alveoli, as I discussed earlier. So the upper part of the lung is what we call proximal. So when I refer to a proximal airway, we're looking at the larger airways, and distal is located down here closer to the alveoli. So there's that difference here. And then the basal cells are located throughout here in the epithelium. So my project specifically is looking at basal cells. So these cells typically express TP63, KRT17, and KRT5. There are certain types of basal cells that don't express KRT5, and they've been identified in the pathogenesis of IPF, which is idiopathic pulmonary fibrosis. However, recent data, RNA sequencing done by HuBMAP actually, has suggested the presence of KRT5 negative cells in healthy human lung tissue, which was pretty abnormal. So that's what we're going to be focusing on now. So now that we know that these negative cells are present in healthy lung tissue, we can look into why they're there and what the function is of them. So in order to do this, we used immunofluorescence and QPAS software in order to identify and quantify the basal cells that were positive for KRT17 and negative for KRT5. And we did this by looking at frozen human lung tissue, and we compared those subsets in proximal and distal lung locations. And we do this because it can help us gain a better understanding of the roles that they play in healthy human lung tissue. Okay, so why do we care? So I talked a little bit about this earlier. So the study of basal cells is a result of the need for different clinical therapies and medication that are caused by either genetic or environmental casualties to the lung epithelium. So diseases such as pulmonary fibrosis, so idiopathic pulmonary fibrosis, acute respiratory distress syndrome, acute lung injury from COVID-19, and cystic fibrosis. So a lot of these diseases are driven partially due to epithelial dysfunctions or some sort of injury done to them and the act of attempting to repair the epithelial layers. So when we're looking at these cells that are negative for KRT5 through single cell analysis, so as I mentioned earlier, there are cells present in healthy human lung tissue that don't contain KRT5, but it hasn't really been thoroughly explored. As I was doing research into the background of basal cells, there's not a super, super large amount of research done on these cells in general, which is interesting since they play a significant role in keeping the lung healthy. So as we can see here, all these different expressions that are expressed in these basal cells, but KRT5 isn't found. So we focused on the basal cell group from the HuBMAP data, And if you look at this violin plot right here, so you can see the presence of TP63, KRT17, but there's a lack of presence of KRT5 here, which is what we're looking at. And again, this is in healthy tissue. So the purpose of the project was to confirm the presence of KRT5 in KRT5 negative basal cells in healthy human lung tissue. And then we're going to analyze the difference in proximal and distal pathways, and then hopefully utilize this information to identify different functional differences and similarities, and we can understand the healthy human lung a little bit better. So we did this by immunofluorescence staining. So the purpose of it, it allows us to get a visual of the localization of these cells. So it's done basically through specific antibodies. We used indirect staining. So this is basically where a primary antibody is attached to the target antigen that we're looking for, and then we use a secondary antibody to attach to that primary antibody, which is tagged by a fluorophore, which allows us to get a good image here. So this is kind of a zoomed out example right here of what we see. Right here, we see two different airways, and everything that lit up here is essentially a basal cell. And the second part of this is HuBMAP. So HuBMAP is a bioimaging software that's used for whole slide scanning and image analysis, and it kind of allows us to get a quantitative assessment of what's going on here. So we can go through and we can single out every individual cell and see whether they're positive for KRT17, KRT5, a double positive if they're co-localized, or a positive for one or the other. So it gives us a really good numerical value of what's going on that particular tissue. So we can see here an image that we took. So up here we have the proximal airway, and it's usually larger, and a distal airway, which is much smaller. And here, if we zoom in, we used a DAPI stain, which is a nuclear stain. And then for KRT17 right here, we have the red. So these are the cells that we're looking for that are positive for 17, but negative for 5. The green right here are the ones that are expressing KRT5. And then wherever you see yellow is co-localization. So cells that express both. So after going through quantification and kind of telling HuBMAP what we were looking for, we noticed significantly higher amounts of KRT5 negative basal cells in distal airways as compared to proximal airways. So we took all these slides for one particular donor. So in the future, I think it would be wise to kind of look at different donors as well, and get different frozen tissue from there. So in summary, we essentially confirmed the presence of the KRT5 negative basal cells in the airway, which confirms the data that was collected earlier from HuBMAP. And the quantitative image analysis demonstrated a statistically significantly higher percentage of these KRT5 negative cells in distal airways compared to the proximal. So we know that these cells could play a crucial role in maintaining homeostasis in the lung, and not just the pathogenesis of IPF. So moving forward, it would definitely be cool to kind of look into differences between a healthy lung and a diseased lung, and then kind of looking into these further subsets of the other basal cells, because there's different types of basal cells, different subsets, and what exactly their functions are. Why are some expressing 17? Why are some not? Why are some expressing 5? And why others aren't either? And why is there a difference in the proximal and distal airways? So further investigation into the functions and localizations of these cells could give us insights into applications and therapeutic interventions for lung diseases caused ultimately by epithelial dysfunction. So some takeaways from my experience overall, I've definitely learned that research is not a linear process. When we're in school, we learn about a scientific method. We have a question, a hypothesis, we do the experiment, and we look to get an answer. It's not always like that. Sometimes we end up with more questions than what we began with, as I found myself here. So we understand that there's a higher presence of these cells in the distal airway, but why is that? Why do we find that? What role does it play? and that mistakes happen in the lab, especially if you're new to the lab, and even experienced people come across these same issues, but ultimately makes you better. And you can learn from it, which brings me to my next point, that it was a constant learning experience. I felt like I was learning something new every day, which is great because you never know everything about what you're working on. You're doing the research to gain more information, and it's a collaborative effort with everyone in the lab and different labs reading up on research and data found from other people outside the institution. So I'd like to thank everybody in my lab, Dr. James Haygood, for giving me the opportunity, and everyone else that kind of collaborated with me, the NIH and HuBMAP itself, for giving me the opportunity to intern here, and the University of North Carolina at Chapel Hill for all the resources provided and guiding me in the right direction as I move through this project. And if anyone has any questions, I'd be open to answering them.